Namaskara Karnataka, I am Miss Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is visible. Bega comment ಮಾಡಿ chats ಅಲ್ಲಿ if I am audible and visible to all of you. ಹೇಗಿದ್ದೀರಾ ಎಲ್ಲರೂ? ತುಂಬ ದಿನ ಆಯ್ತಲ್ವಾ ನಾವು ಮೀಟ್ ಮಾಡಿ. Yes, tell me, are we live? And also let me inform on Telegram as well for students to join. Right behind me is visible. Yes, we are audible and visible. Now let's just inform on Telegram. Yes. Are you all ready for it? Hello. Yes, quickly like the video as well, okay? Bega like mari video na, so that we can move on with a very energetic class, that is your molecular basis of inheritance. Now for those students who do not know anything about molecular basis of inheritance, do not worry at all. I am going to teach you everything, right? Molecular basis of inheritance, I am going to divide it into multiple parts, because in one shot if I try to do it, it will take me at least 5 to 6 hours. So instead of that, we are going to do small kuti portions of it, and trust me, no more this chapter is going to be a difficult thing for you. I am going to make you understand every line of NCERT and also make you understand the important concepts for your KCET as well as board's point of view. Okay, ready na ellaru? Very nice. So molecular basis of inheritance, students, has the highest weightage in any competitive exam for that matter. So here also their weightage is around 4 to 6 questions in KCET. So can you skip this chapter? Of course not, okay? Imagine it, even in some places you will see up to 8 questions also come, but 6 questions max from this topic is pakka. Now which topic, which question is going to come, we cannot predict. So what we are supposed to do? We are supposed to learn everything, right? Ready? Yes, okay. So today I am going to teach you what is DNA, what is RNA, what is the structure of DNA, how does the bonding happen, and the different experimental setup. So look here students, DNA is called a nucleic acid, but how did this whole thing come into place? Now when we look, right, when we look at let's say you went to a relative's house and they had a just born baby. Now when you look at the just born baby's face, you feel very cute, right? You will be like, oh my god, very cute, yeah, baby is. Now you will compare the baby's face. Some people, like the older people, will compare the baby's face with the mother, with the father. But how is all this genetic information stored, right? All our genetic information is stored in the form of our DNA, right? It is a small portion, again gene we tell, right? A small portion in the DNA or a small segment in the DNA is called a gene. Now all this inheritance, all the information that is going to code for the proteins is stored in the DNA. So is DNA a simple person? No. DNA is a very hi-fi person, right? Because it is going to store all the informations required for you to, carry forward, right? Generation after generation, we will notice that some things in our family is carried forward, right? Now DNA finding, okay, how this whole thing came into existence, students. DNA, or this nucleic acid word, came from a scientist called as Friedrich Mischer. So he is told that he was working on pus cells, and when he was working on that, by fluke, by chance we tell, no, ayo, by chance engo pass agidini. So we tell it very casually, like that only. By chance Friedrich Mischer found something called as nuclein, okay. So he found and he called it as nuclein in 1869. So DNA is an acidic substance present in the nucleus, and it was first identified by Friedrich Mischer. Important from NCERT point of view, it is given in your NCERT. Now students, he found that it was, okay, let's call it nuclein. Then there is another scientist whose name was Altman. What was this scientist's name? Altman. A-L-T man. Altman told that the nuclein that you found, right, the nuclein that Friedrich Mischer found has an acidic nature. Has an what nature? Acidic nature. Students, energy is not enough. Tumba kadme agide energy. Okay, I want all of you to pick up your energy and tell, yes ma'am, answer all the questions, and also share the video with your friends. Okay, share the video with your friends. Let me also share it. I think I forgot to share it here. Yes, done. Okay. Fine. So, Altman told that nuclein which Friedrich Mischer found has an acidic nature. So, we will not call it anymore nuclein, but we will call it what? We will call this nucleic acid. We will call this what? Nucleic acid. Now, do you know where will you get all these notes? You will get it on Vedantu, KCET and boards channel, and if you want to join my channel, it is biology by Gopika. So, nucleic acid, okay. So, Altman told that nuclein had an acidic nature, so we will call it nucleic acid, and later the two nucleic acids that we found was what? DNA and RNA. But tell me, what is the full form? DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, and RNA stands for ribonucleic acid. Ribonucleic acid. So, when you look at the naming itself, all of you understand that there is something called as deoxyribonucleic acid and ribonucleic acid. Ma'am, what is this deoxy? Deoxy means somewhere, in some position on the carbon, on the sugar, one oxygen is not there. But this fellow has that oxygen. That is from where the term comes, deoxy. That means absence of an oxygen group. Absence of oxygen. And do you think it is a disadvantage or an advantage for this, a DNA? I would say it is an advantage for DNA, because when you learn the properties of a genetic material, some properties of genetic material is that it should be replicating, it should do mutation, but at a very slower process, and also it should be structurally and chemically stable. But this fellow has an extra oxygen. Because of this, he is reactive, full time active he is. He'll go and bind with them, he'll go. So it is fully, a mess. But this RNA, because of the presence of an oxygen, is a mess, but in another place, that absence of an oxygen made DNA the most stable nucleic acid. Is this clear to all of you? So, Altman told nuclein has an acidic nature, so we came up with the word nucleic acid. Two types of nucleic acid we know, DNA and RNA. Now, 99% of the organisms, right, majority of the organisms has DNA as the genetic material, right? Most organisms possesses DNA as the genetic material. Ma'am, then what about RNA? RNA also is genetic material, students, but in some viruses. In what? Some viruses. You want example? Like tobacco mosaic virus. Okay? Tobacco mosaic virus. TMV, tobacco mosaic virus. Now, DNA is an acidic substance, all of this is clear. So, this is your introduction to the chapter. Now, let's move further and let's understand more about DNA, because your focus should also be in understanding DNA. Now, we know this is deoxyribonucleic acid. Students, DNA or the genetic material as a whole, okay, nucleic acid is made up of units, okay. So, let's take an example of starch. Starch, I am telling, it is made up of multiple units, okay, it is made up of many units of what? Glucose, like that, glucose, many monomers are coming, right, many monomers. So, we will call this monomers. Like that, our genetic material also is formed out of, units which are called as nucleotides. So, DNA is made of nucleotides. So, many nucleotides, right? So, let's say nucleotide, I am writing NUC, like that, many nucleotides together form the DNA. So, we can also call this as a polynucleotide chain. Polynucleotide chain. Clear? Polynucleotide chain. Artha aita ellargu? Polynucleotide chain. So, that is why in your textbook you will see structure of polynucleotide chain. For those students who don't understand, they'll be like, my God, What is this? But do you understand? Like how starch is made up of multiple glucose. Okay, multiple glucose. Like that DNA also is made up of many units of nucleotide. Together we call it the polynucleotide. So what structure you are seeing here is nucleotide, nucleotide, nucleotide bonding with each other to form the DNA structure. Now, in 1983, okay, very important, in 1983, James Watson and Francis Crick, based on an X-ray diffraction data, found that with the help of Maurice Wilkins and Rosalind Franklin, proposed that, very simple, that DNA is a double helical model. That means what, students? If I see a DNA, I will notice that DNA is a double stranded structure, like this. Double stranded structure. And if I zoom further, okay, if I zoom further, each portion I am going to see a nucleotide. Okay, so let's say many nucleotides come together and one is going to form the backbone and another is going to be in the between. Let's say this is your DNA. Okay, this is one strand, this is one strand, and they both are not going to, they are going to run anti-parallel. One is from 5' to 3', another one is from 3' to 5'. Okay, cleared all of you? Now, let's understand what is this nucleotide made up of. Now, nucleotide, I told you, it is made up of multiple compounds, right? Multiple compounds. Now, nucleotide is made up of 3 compounds. Okay, components. What are these 3 components, ma'am? First, it is made up of a sugar molecule. Then it is made up of a phosphate molecule. Okay, no, wait, let's write first nitrogen bases so I can teach you one more topic like this. Nitrogenous bases. Another one, it's made up of phosphate group. So what did I say? I told you that DNA is made up of many nucleotides coming together, and many nucleotides coming together, right? Many nucleotides coming together, it is going to form a DNA structure. And I am also telling you that nucleotides are made up of 3 components, which are sugar, nitrogenous base and phosphate group. Now all of you have to remember one word, sugar plus nitrogenous base, okay? Sugar plus nitrogenous base together we can call nucleoside. We can call what? Nucleoside, because you are pushing one person to the side. Whom are you pushing to one side? You are pushing the phosphate group to one side. So sugar plus nitrogenous base together you can call it a nucleoside because you are pushing phosphate to one side. But ma'am, 3 together, is it nucleotide? Yes, definitely. All 3 together, right? All 3 together is a nucleotide. Done? Cleared all of you? Okay, it's a nucleotide. Yes, it's a nucleotide. Now, the most important thing is, what is this each structure when we talk about, right? So, let's start with sugar. Let's start with sugar. So, students, 2 types of sugars are there. Ribose sugar. Ribose sugar. This is a very, very important chapter. So, those who are watching it later recorded also, let me know if it was helpful or not. Okay. So, ribose sugar and deoxyribose sugar. Deoxyribose sugar. So, ribose sugar, deoxyribose sugar means what? In ribose sugar, in the second carbon, okay, second carbon, there is going to be an OH, and here the second carbon will not have an OH, it will just have a H. This makes these 2 very, very different, okay. Clear to all of you? This is the reason why DNA. So, DNA will have deoxyribose sugar. Yes, ma'am, no doubts in that. So, this is going to be possessed by DNA. This is going to be possessed by RNA. Okay, RNA. So, RNA has an extra OH group, O group in the second carbon, and that will not have, that is deoxyribose sugar will not have. Now, the next topic that comes is nitrogen bases. So, nitrogen bases, when we talk about, students, remember that nitrogen bases are mainly divided into 2 categories. I think I will write it in the next sheet. By then I will show you phosphate group. So, phosphate group is very simple. Okay, phosphate group is very simple. A P with double bond O, O, O and O. Okay, 4 oxygens combined on the phosphate group. Okay, clear to all of you? So this is your phosphate group. Now when I have to talk about nitrogen bases, it is a more in-depth topic. Okay, it is a more in-depth topic. So I'll show you the bonding as well. Now listen to me carefully. When we talk about nitrogen bases, remember that this is how your DNA is going to be. Okay, now let's say I unwound the DNA, or first let's draw this. Okay, this is your DNA structure. Let's say I unwound the DNA like this and like this. Okay, I unwound the DNA. Now what you are supposed to remember? You are supposed to remember that the backbone of the DNA means what? This structure is made up of sugar and phosphate, and in the middle, to hold the structure together, we have the nitrogen bases. So if I untangle the DNA like this, I think I got tangled inside the wire. Okay, if I untangle the DNA like this is going to be one base. Okay, let's draw like, okay, like this. This is how your bases are going to be arranged. Okay, same thing on the other side, and between them hydrogen bond is going to be there. Okay, hydrogen bond is going to be there. Clear? So the bases are going to project inwards. Okay, hi Baswana. It is going to project inwards. So remember that nitrogen bases are mainly divided into two categories, forming purines and pyrimidines. Purines and pyrimidines. So there is a way to remember, students. In purines, two of them is going to come, that is your adenine and guanine. Okay, and in pyrimidines, three are going to come. Remember, cut: cytosine, uracil and thymine. Uracil and thymine. Clear? Yes. Now ma'am, how do I remember this? It's too difficult for me to remember. So in purines, you are going to remember this way: pure as gold. What are you going to remember? Pure as gold. So P for purine, A for adenine, G for guanine. Now how are you going to remember pyrimidines? You are going to remember cut. C-U-T. Cut. Okay, cytosine, uracil and thymine. Now very catchy thing about this is, ma'am, how can one have three, one have two? Here, uracil is only present in RNA and thymine is only present in DNA. Okay, this is also present in DNA. This is also present in DNA. So that means what I am trying to say is, instead of thymine, okay, instead of thymine, uracil is going to be present. So uracil is found in RNA. That is the reason why we have three of them. Clear to all of you? So ma'am, now what is the whole structure like, right? We have sugar, we have. So students basically understand when I am drawing it. Okay, let's draw one beautiful diagram for it for all of you to understand. So this is how your DNA molecule, okay? This is your phosphate group. Okay, this is your phosphate group. Clear? The red dot is your phosphate group. Now this is the bond that is going to be formed. Okay, then we have to draw a sugar group. Okay, one second. Okay, a sugar. So this is going to be your P, this is going to be the sugar, and then that sugar attached. Okay, I think I should draw the sugar with a different color. So phosphate group is confirmed, no ma'am? Which lecture, dear? Which are you talking about? Are you talking about sexual reproduction, or are you talking about something else? I did not get you. Just tell me Okay, let's draw the bonds all with white, okay? Let's draw all the bonds with white. Now, let's draw the sugar molecule, which will have a O. Okay, this is how it is going to be. Now, students, the problem is I am not supposed to draw the bond. I am supposed to draw the bond with the, with the first carbon. So, that's why it's taking some time, okay, because beauty matters. Yes, perfect. Yes, we are done with a simple structure of how the entire DNA molecule is going to look. Yes, so look here. Yes, this is a structure, okay, simple structure. This is many of them like this will be there. Now, what you are supposed to remember is that what I have drawn here is a deoxyribose sugar. Okay, this is the deoxyribose sugar, and you will get all these notes, don't worry. Okay, and this which I have drawn here is the base. Okay, is the base, any nitrogen base. It could be an adenine or it could be a purine, and here we have the sugar molecule. Okay, this is the sugar and this is the phosphate. This is the phosphate. Clear? So, this is how a simple section, sorry, because you asked me sexual reproduction. Okay, this is a simple how your nucleotides are going to be arranged. Now, remember that your purines are actually, purine, small word, no, but it's big. Okay, purines are a 9-ringed structure. A 9-ringed structure, okay, but pyrimidines are a 6-ringed structure. Okay, 6-ringed structure. So, this much you can remember, students, about your nitrogen base. Okay, sexual reproduction, flowering plants, I will only be picking the most important topics. So, just let me know which are the topics, because chapter wise if I go, it will take a lot of time. We will definitely do chapter wise, but just tell me how which topic you are finding it difficult. Now, students, since you see this, remember that your sugar, okay, very important, remember that your sugar plus phosphate, sugar plus phosphate is going to make the backbone of DNA. What is it going to make? The backbone of DNA. Ma'am, then what about nitrogen bases? They are going to project inwards like this. They are going to project inwards with the help of hydrogen bonds, so that they can make sure that the DNA structure stays in place. Okay, DNA structure stays in place. Now, remember, purines and pyrimidines are always going to be in same numbers. Why? Because it is proven by Chargaff. The scientist's name is Chargaff. He told that the ratio of purines and pyrimidines is always going to be equal. That means purines, ratio of purines should always be equal to the ratio of pyrimidines. That simply means that they are going to bind or bound with each other. Now, how do you remember that? You are going to use this trick, students. The trick is at Gopika ma'am's class. At Gopika ma'am's class. Okay, so A is going to bind with T, G is going to bind with C. A is going to bind with T with 2 hydrogen bonds, and C is going to bind with G is going to bind with C with 3 hydrogen bonds. Can you remember this? Okay, Chargaff rule, can you remember? Yes, sure, I'll take it up. Zainab, I will take it up. Okay, don't worry, we'll definitely do that. Yes, students, is this clear? So, Chargaff told that how many purines are there, that many pyrimidines should be there. Like how we tell, no, when you are making a group in the class, how many boys are there, that many girls should be there. So, we can make pairs for a dance, annual day dance. Same way here also, how many purines are there, that many pyrimidines should be there. Now, how do they bind, ma'am? They are going to bind with this formula that adenine always binds with thymine and guanine will always bind with cytosine. How will you remember? My name. At Gopika ma'am's class. Ma'am, you keep it in mind. So, at Gopika's class, A will bind with T, G will bind with C. Clear? Now, you have to know that how many bondings are they going to bind and what are the bonds that are formed. So, I have drawn a very complicated structure here, but I am going to simplify it and tell it to all of you. Okay, look here, students. This is what? This is a purine, a 9-ringed structure, okay, a 9-ring structure. A 9-ring structure is always going to bind, okay, a nitrogen base is always going to bind to the first carbon on the sugar. This is what? A sugar molecule. So, on the first carbon, it is going to bind, and that bond is called as N-glycosidic linkage. See, if you do not listen to the class carefully, this chapter will always remain difficult for you. Listen to me carefully. It is a very simple topic. See, the nitrogen base is going to bind with the carbon in the first position. Okay, and that bond formation is called as N-glycosidic linkage. Clear? Now, to the sugar, phosphate is going to bind to the 5th carbon. Phosphate is going to bind with the 5th carbon here, which is called a phosphoester bond, with the help of phosphoester bond. Now, one nucleotide, this is one nucleotide, this is another nucleotide. They both are going to bind to each other with the help of a phosphodiester bond. Why is it di? Because there are two nucleotides. This is one nucleotide, this is one nucleotide. These two will bind with the help of phosphodiester bond. Sugar will bind with nitrogen base will bind with sugar with N-glycosidic linkage. To the sugar, phosphate will bind with phosphoester linkage. Is this clear to all of you? Yes or no? Now, remember that in your entire DNA structure, if you go back to this diagram given by your NCERT, all of you will notice one thing, that this here 5 carbon of phosphate is empty. Okay, here 5 carbon phosphate is empty. Here on the 3 carbon, this is how this is ulta given like this, okay. The 3 carbon OH is empty. So, we will call this angle as phi dash to 3 dash. Now, this way you see, here again 3 OH is free, there 5 phosphate is free. This will be called from 3 dash to 5 dash. So, this is how the polarity of the strands will be found. That means one side 5 phosphate will be free, one side 3 OH will be free. This side 5 phosphate will be free, this side 3 OH will be free. So, this will lead to the binding of the next one, no? So, it will be called as 5 dash to 3 dash, 3 dash to 5 dash. Clear? Students, I know in the first time this chapter is not going to sound easier, okay, it will take multiple revisions for you to learn this chapter thoroughly, but it is okay, do not give up, it is a very important chapter, so you have to make a point to learn it however difficult it is, okay. So, here what is the bonding that you have to remember? You have to remember that a nitrogen base NB will bind with a sugar with N-glycosidic linkage. At which position on the sugar? At the first position of the sugar. Clear? Now, the sugar, to the sugar, phosphate is going to bind, that is PO4 is going to bind to the 5th carbon. Can you see here? I have labeled 5th carbon, CH2OH is going to bind, and this is going to happen with a phosphoester linkage. Okay, phosphoester linkage or bond, you can use the word. Now, when two nucleotides have to bind together, they will bind with phosphodiester linkage. Okay, so 5 dash to 3 dash, 3 dash to 5 dash. So, that's why we tell they are anti-parallel. Okay, they are not coming in same polarity, right? It's not 5 dash, 5 dash. Okay, it is 5 dash to 3 dash, 3 dash to 5 dash. Is this clear? Linkage is clear? So, here you can see I have put the salient features of DNA. Now, let's do the salient features. I hope the bonding is very clear. Now, let me tell you one additional information. One second. Yes, I am taking this slide. Okay. See, students, now some additional, not additional, like important information. First of all, remember, one sugar is there. Okay, one sugar is there. To the sugar, a nitrogen base has to come and bind. Okay, NB has to come and bind anything. It could be a purine or it could be a pyrimidine. Now, purine, it can be adenine on guanine, and purine can be adenine and guanine. Pyrimidine can be cytosine and thymine, because we are talking about DNA. So, if these both has to bind, it is 100% sure that they will bind with N-glycosidic linkage. N-glycosidic linkage. Yes or no? Now to this sugar molecule, let's say one phosphate group on the 5th carbon has to come and bind, that is a PO4. Okay, 5th carbon has to bind. That will happen with the help of phosphoester linkage. Yes? Now this is an entire nucleotide. Okay, this is an entire nucleotide. So let's just border this entire nucleotide. Okay, to this nucleotide, another nucleotide wants to come and bind. Okay, one more nucleotide wants to come and bind this with another nucleotide. That will lead to what bond? That will lead to a phosphodiester bond. Phosphodiester bond. Okay, remember this is clear. Now remember that in the 5th carbon, okay, 5th carbon, one phosphate will be free, 5 dash. Okay, and in the same DNA, in the 3 dash, there will be an OH free. Okay, OH free. So we will call this DNA strands as 5 dash to 3 dash. 5 dash to 3 dash. Now the next strand, how is the next strand going to be? The next strand will have OH, 3 dash OH free, and let's move forward, it will have a P group with 5 dash, right? So I'll write it down. So here we will say that 3 dash to 5 dash. This is how the both strand polarity is going to be. Okay, it's very important to check the polarity and understand the directionality. Okay. So understand this. Now I told you one more thing about the Chargaff rule. So remember students, Chargaff rule spoke about how the ratio, right, how the ratio of purines should be equal. Should I write it separately? One second. Yes. How the ratio of purines should be equal to the ratio of pyrimidine. Okay, that means how many A's are there? Okay, let's take an example. A is 30%. A is what? Adenine. Okay, I'm writing adenine. Adenine is 30%. Then how many guanine should be there? All of you tell me in the chat box. If adenine is 30%, sorry, how many thymine should be there, because adenine is going to bind with thymine? How many thymine should be there? All of you tell me in the chat. Quickly answer. Yes. All of you know the answer. Come on. See, adenine is 30%. That means adenine is going to bind with thymine. At Gopika ma'am's class, adenine is going to bind with thymine. So, I would say that thymine should also be 30%. If thymine and thymine, purine, very good. Kush Mohita, very good. If adenine and thymine is 30-30, then what is left out of 100? We have 40%. Then your guanine will be 20%. How much will be cytosine? How much will be cytosine? Can you answer? Cytosine, how much it will be? So, a total of 100 happened. So, Chargaff told that how much adenine is there? How much purines is there? That many pyrimidines should be there, right? Then only binding will happen. Now, how do their binding happen? We know that their binding is going to happen, that adenine is going to bind with thymine with 2 hydrogen bonds. 2 hydrogen bonds I have written here, okay? And the cytosine is going to bind with, guanine is going to bind with cytosine with 3 hydrogen bonds, okay? So, hydrogen bonds are a medium of binding here. Done? Done? Yes, very good. 20%, right? 20% of cytosine should be there. 20% of cytosine should be there. Clear? Very good. Understood? So, that is the Chargaff rule talking about. Okay? And understood, no? They are going to bind with the help of hydrogen bonds. Hydrogen bonds. This is the whole thing. Binding, you remember students, okay, how the bonds are going to happen and how they are going to bind with the next nucleotide, very important. Bonding you know, polarity you know, then you are done, okay, and then Chargaff rule. Now let's understand the salient features, okay, let's understand the salient features of DNA. What are the salient features of DNA, okay? Now, the salient features of the double helical structure of DNA are followed. It is made up of two polynucleotide chain. We know what is nucleotide. We know many nucleotide together will form a polynucleotide chain, and we know it's a double helical structure. One strand here going around each other. So, two polynucleotide chain where the backbone is constituted by a sugar and phosphate, right? And the bases project inwards. Understood? So, this is the two bond. This is made up of sugar and phosphate. In between them, your bases are going to be there. Okay. Now, the two chains have anti-parallel polarity. It means if the chain has 5' to 3', the next chain will have 3' to 5'. This is also clear. The polarity is going to be anti-parallel. The bases in two strands are paired through hydrogen bond. The bases are bound with hydrogen bonds, okay, forming base pairs. So, we can call them pairs, no? Like if marriage happened, then we will call them, no, couples, okay, pairs we tell them, no? So, like that only. So, here what has happened? Bonding has happened. So, obviously we call them base pairs. Adenine forms two hydrogens with thymine from the opposite strand and vice versa, and guanine is bonded with cytosine with 3 hydrogen bonds. Tell me who is stronger in this, you know, in this marriage, who is stronger couple? Is it adenine and thymine or cytosine and guanine? All of you tell me answer. In this, who is the stronger couple? Who is the stronger couple? Is it adenine and thymine or cytosine and guanine? Yes, quickly answer. You people have to pick up your speed, okay, in typing. You are slow in typing. Tell quickly. Who is stronger couple? Adenine, thymine, cytosine, guanine. Ma'am, obviously understood, ma'am. It is guanine and cytosine, right? Guanine and cytosine because they are bounded with 3 hydrogen bonds. As a result, always a purine comes opposite to the pyrimidine. This generates approximately uniform distance between two strands, okay? They are going to come against each other, no? They are going to have uniform, distance. The two chains are coiled in a right-handed fashion. Very important. The pitch of the helix, that means in one turn, how much it is going to be? 3.4 nanometers. So, let's say this is one strand, one turn happens, okay, like this one turn happens, it's going to be the pitch, 3.4 nanometer. A nanometer is 1/1000th of the meter, so that is around 10 to the power minus 9 meters. And there is roughly 10 base pairs, okay, 10 base pairs in each of the turns. Consequently, the distance between a base pair in a helix is approximately 0.34. So, you can do this calculation, no? See, we know that one pitch 3.4 we know. And I told you in one pitch there are around 10 base pairs. So, what will be the distance between them? 0.34. 0.34. Clear? Pitch and the distance of the base pairs. Now, the plane of one base pair stacks over the other in the double helical. This gives more stability. That means one base pair over the other, they are going to stack with each other. So, the bonding or the stability between them is very, very stronger. Clear? Salient features are clear? So, students, here you can see in the DNA there are minor grooves and major grooves. Major grooves have the sequence places, minor grooves have the non-sequence places. Here the proteins will be able to come in mind. That's a different story by itself. But remember like this is the backbone which is made up of sugar and phosphate. This backbone which you are seeing, no, it's made up of sugar plus phosphate, S plus P. And what you are seeing in the between is your base pairs. okay, which is going to help in the stability. And you see one turn, okay, will be 3.4 nanometer, and in one turn how many base pairs will be there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pairs, okay, 10 base pairs will be there in one turn. In one turn. Technically this diagram will not prove it, but in one turn you will see 10 base pairs, okay, clear? Salient features are clear. Can we go to packaging of DNA? I have taught this before also to all of you. Remember that the nucleus is a kutti one, right? So where do we find our DNA? Inside the nucleus. Okay, this is the cell. Okay, this is the cell. Inside the cell we have a kutti nucleus. Okay, cell is dancing, students. Like madidira ellaru? Did you like? Okay, did you like the video and mark your attendance? Then only I will know that oh, this student was there in the class. So here, this is the nucleus. This is a kutti nucleus which has around 10 to the power minus 6. It is very small nucleus. But my DNA which has to get inside the nucleus is a total of 2.2 meters. Okay, how much? 2.2 meters long, approximately. Now, how will I make this 2.2 meter long DNA inside this nucleus? Not possible, ma'am. You just leave this idea. This is not possible. No, it is possible. For that, I am going to take the help of some proteins which are called the histone proteins. Histone proteins. See, we know that DNA is negatively charged, right? This fellow, DNA, is negatively charged. So, I am pakka sure that if the protein is positively charged, then the DNA is going to get attracted. So, these histone proteins are going to make a structure that has 8 proteins, and we will call this as histone octamer. We are going to call this as what? Histone octamer. This histone octamer, around this histone octamer, I am going to wound my DNA around it. So, let us say this is my DNA, okay. To this DNA, I am going to wound around my histone proteins, histone octamers, okay, and my DNA is going to go around again, right. It is going to wound around and it's going to continue like this. So, like this my entire combination is going to happen. Clear to all of you, right? This entire combination is going to happen. Now, ma'am, do they have a name? Yes, they do have a name. Histone octamer plus DNA, okay. So, listen to me carefully. Histone octamer plus DNA will be together known as the nucleosome. Together will be known as the nucleosome. Okay, around 200 nucleosomes. Okay, base pairs will be there in a nucleosome. So here you can remember that 200 histone octamer plus DNA is equal to nucleosome, right? Like this, many nucleosomes come together to form your chromatin fibers. Like that, it becomes the chromosome. So now, students, remember that this histone octamer is not just made up of one protein. Okay, there are around 5. Okay, 5 people who are going to help for this. Who are the 5 people is the question. H2A, H2B, H3, H4, and at the last in your NCERT there is one more protein given as H1 protein, which is also called the linker protein. This linker protein is going to do what? It's going to link everything and keep together, no? So it's going to do the linking job. So let's say, let's draw a Kuti structure. This is your histone H2A. Okay, this is H2A. This is H2B. Okay, this is H2B. This is H3. Okay, H3, and let's say this is H4. Okay, so can you see they have formed an octamer, not just one. Okay, not just one, they have formed 8 of them. Okay, 8 of them. Yes, Mintu ma'am. Okay, so they have formed 8 of them, students. So this is going to be my H2A, H2A, H2B, H3, H4. This together has formed the octamer, 2 each. Okay, H2A, H2B, 2 each have formed an octamer. Now around, to keep the DNA, obviously the DNA is going to wound around it, right? The DNA is going to go like this, it's going to go like this, come out like this. Okay, it's going to wound around the whole thing. To keep all of this in place, okay, to keep all of this in place, we have a protein which is called as the linker protein. What do we have? A linker protein or the H1 protein. H1 protein. In your DNA, we will in your NCERT you will see linker protein. Clear? Any doubts on this? No doubts, right? Perfectly clear to all of you? So this is how your entire thing happens, and here I have put a diagram which I only drew, okay, to make you understand. So students, this is how your DNA strands were. They wound around the histone octamers. So around, see, I told you nucleosomes have 200 base pairs. It went around the histone octamers, and the orange color that you're seeing is your H1 protein. Like this it went, again it rolled, like this it became the chromatin, again it went to condensation and then formed your chromosomes. So when you're doing your prophase, metaphase, anaphase, telophase in the labs, right, when in the root tip, with the help of root tip, you would have noticed that there are some, you know, mixed structures, right? Some thread-like structures. What are they? They are your chromatin fibers. Then they condense more to form your chromosomes, which you will be able to see in the metaphase. Then you will see the separation of the sister chromatids in the anaphase, right? So all this what you were seeing is actually the binding. Okay, clear? Now remember that lysine and arginine is the most common amino acids seen in the histone is a protein. Protein is made up of amino acids. So the most common amino acids are your lysine and arginine. Okay, amino acids are lysine and arginine. Okay, this question can come as an MCQ, so remember this very, very importantly. Okay, the diagram is clear. DNA was there, which was 2.2 meters. It wouldn't fit into the nucleus, so we took the help of proteins which are called as histone octamers, okay, histones, and they formed octamer structure. So this is an entire octamer. Okay, with the DNA we will call it a nucleosome, and then we have a linker protein which is the H1 protein, and they will come together by condensation, again condense to form the chromatin. Okay, chromatin fibers. From there again condensation will happen, chromosomes will happen, and if you see it under the microscope, you will see it as a bead on a string structure. Okay, you will feel like one bead is there, one string is there, one bead is there, like how they put this mala, students. Okay, they make this mala, no, they make a jasmine one, they make different types of chain. You will notice that they tie a knot as they move forward by keeping the flowers. Why? So that the flowers do not move. That's it. This side. Same way linker protein is going to do that job. Okay, now NHC is non-histone chromosomal protein, which are also used for the same condensation and, you know, packaging only, but they are used in higher organisms. Clear? Like in the higher coiling, when we need higher coiling, additional set of proteins when we need, we are going to use them. Any doubts in this? We are going to start the experiments. Okay, so packaging is done, structure of DNA is done, binding is done, what is nucleic acid is done, all this is done. Now we will go into the experiment. So the first experiment that came into existence to tell that DNA is there or something is there was Griffith's experiment. Okay, first experiment given is your Griffith's experiment. In the Griffith's experiment, you have to remember that he was working on Streptococcus pneumoniae. Coccus means shape, you know, spherical. Pneumoniae. Streptococcus pneumoniae, which causes pneumonia. When he was working on Streptococcus pneumoniae, he observed that these have 2 strains. Okay, many strains can be there, students. When you go to lab, you will understand many strains were there. But Streptococcus pneumoniae, he noticed 2 strains were there. One was called the R strain or the rough strain, and one was called the smooth strain or the S strain. This is the S and this was denoted by the R. Now, you might ask me a doubt, ma'am, how? What was the difference? So, in the rough strain, he noticed, okay, a rough strain was there, and then he noticed the smooth strain. And he thought that the smooth strain is a smooth strain because of the presence of a mucopolysaccharide which was covering the smooth strain. Okay, so this outside coating was done by a mucopolysaccharide. This was absent in this rough strain, which would have made the rough strain structure like this. Okay, as you can see here, this is your rough strain and this is your smooth strain, and the smooth strain had a capsule. You can either call it a capsule or you can call it a mucopolysaccharide. Okay, there was a capsule which was there, and this capsule would have made the smooth strain virulent. Virulent means what, ma'am? Virulent andre the disease causing one. Virulent means the dangerous, no, the villain type of person. Here it is the disease causing one, and this was non-virulent power. Okay, non-virulent. Okay, non-virulent. So, rough strain was there, smooth strain was there. He was working on Streptococcus pneumoniae, and when he was working on Streptococcus pneumoniae, he saw two strains. One was R strain, one was S strain. S strain, he noticed that there is a capsule layer which is not there in the rough strain, and because of which maybe S strain is virulent. Now, the story starts here when he started injecting that into the mice. So, first he took the S strain, he injected into the mice. The mice obviously dies. No doubt in this. We already know that the S strain is virulent, disease causing, dangerous fellow. Obviously, if you inject it, the person is going to die. No? So, like that, here the mice died. Now, when we injected the R strain, the mice lives. Obviously, because of the fact that R strain is non-virulent. Right. This was very clear, students. There was no magic here. This fellow was virulent. So, the mice died. This fellow was non-virulent. Then he thought, let me take the experiment to a next level. What he took? He heat killed the S strain. He gave maximum heat and he tried to kill the S strain, and he injected into the mice and the mice lived. Okay, he injected into the mice and mice lived. Now he thought one thing. Let me do one thing. Two scenarios where the mice lived. That is scenario number 1 and scenario number 2. I will mix this scenario 1 and 2. That is, wherever situation the mice was able to survive, I am going to take that together and I am going to do something. That is, he took heat killed S strain and he took live R strain. He injected into the mice. Unfortunately, the mice died. Now, if you were Griffith, you would have got shocked. Ayyo, yenaitiddu? Alva, you would have asked yourself, no, what happened? Both the scenarios, individually the mice was alive. But when I mixed it, something happened. Even Griffith got full tension. What happened? What happened? Then he told something. Okay, then he told something from heat killed. Something from heat killed S strain transformed to the R strain. That made R strain, okay, that made R strain virulent, leading to the murder, okay, murder of the mice. Is this clear? Is this clear to all of you? So, he told that something got transformed and he does not know what it is. So, he is just going to call that as the transforming principle. So, that is how the word transforming principle came. Transforming principle. Clear? He did not know what got transformed. He did not know. He knew that something got transformed from the S strain, heat killed S strain to the R strain. So, he called it the transforming principle. Then many other zoologists and botanists and everyone came after it and they were trying to investigate. Okay, so this is the scenario, students. R strain non-virulent, S strain virulent. Here the mice lives, here the mice dies. Here he heat killed the smooth strain and because of which the mice lives. Here he used the both combination. Okay, he used combination 1 and he used combination 3 and here he mixed combination 1 and 3. What did he do? He took the rough strain plus the smooth heat kill smooth strain, and he noticed that the mice dies, and because of which he calls it the transforming principle. Transforming principle. Clear? Now, when we go further, before I go further, let me ask you all one question. Which is correct with reference to the transforming principle? Can you all take a minute and answer this question for me? Take a minute and answer this question for me. Which is the correct with reference to the transforming principle? S strain injected into the mice lives. R strain injected into the mice dies. Okay. S strain heat kill injected into the mice dies. S strain plus R strain injected into the mice dies. Tell me which among this? Pradeep K answered first. Very, very good. Ma'am, answer is going to be this. R, right? This is going to be the answer. Clear? This is the answer. Very good. Okay, very good. I'll give you one homework. Okay, just put the option number in the chat box. Okay, this is your homework, and the homework should be in the comments. Let me see who is the one. Okay, the question is, heat killed S strain bacteria when inserted into the mice led to the transformation of R into S, okay, and the death of the mice stayed alive, transforming of R into S and severe illness in the mice, death of the mice. So just see the option, okay, just see the option and tell me which is the answer in the comments. Okay, we will move further to Avery, MacLeod and McCarty's experiment. Avery, MacLeod and McCarty were the next people who came after this, you know, this transforming principle. So here what happened, they took 3, you know, findings. So they were like, see, we know for a fact that it is not going to be anything else. It should be RNA, DNA or protein that has got transformed from the S strain to the R strain. So first what did they do? They took 3 enzymes. Okay, so let's draw all the 3 of them, students. This is your protein. Okay, protein. This is your RNA, and DNA and DNA. Okay, so they took 3 samples of what combination? They took the same combination of heat killed S strain plus R strain combination. Okay, they took the same combination and they used 3 enzymes. So let's see the 3 enzymes that they used. So first they took protease enzyme. Protease, what are these enzymes? These are the enzymes that can digest or degrade the protein. They took protease, and then they noticed that protein, even after, you know, protease being the protein being digested, here also the mice is going to die. Because why? DNA is still there. So no transformation. No transformation. Okay, no transformation happened. Okay, so protein is not the one. Okay, protein is not the one. Let's do one thing. Let's do it here. Okay, protein is not the one, confirmed. Then they did the next set of experiment where they used RNAs. Okay, they took RNAs. Here what is going to happen? RNA is going to get degraded. Here also tell me what could happen, students. Is the mice going to be alive or is the mice going to die? All of you in the chats. Let me see who is the one who understood. And I can't see the likes increasing. Okay, why? All of you are not enjoying? Yes, the answer is going to be, here we used RNAs, RNA got degraded, but still RNA is not the transforming principle. So here also the mice is going to die because DNA is still going to get transformed. Here also no transformation. No transformation. Okay, so then it was proved that it is not protein, it is not RNA. Then the last hope. Okay, no, see, transforming principle we are going to talk about. Okay, so transformation only happens at this stage. That means here 2 stages the mice is going to die. Protein is getting digested by protease, but that is not the transforming principle. So here last when we come, we have here the DNase that is going to be used. DNase is going to be used. DNase is going to digest the DNA. Here what is going to happen? We notice that the mice lives. Okay, mice lives. That means here this becomes the transforming principle. Clear? Yes? No transformation here. No transformation here. Finally, transforming principle becomes your DNase. Clear to all of you? So, this is what your Avery, MacLeod and McCarty proved, that DNA is the genetic material. Sorry, DNA is the transforming principle. DNA is the transforming principle. But students, this experiment did not go for longer. This experiment failed because many people were not convinced how this experiment was put across. There's a lot of confusion in this experiment, but I hope you understood the whole concept, right? Now, then the final and the last most proven experiment was given by Hershey and Chase, okay, where they took a bacteriophage. Bacteriophage is a virus, okay, remember it is a virus that infects bacteria. Okay, that infects bacteria. Virus that infects bacteria is called a bacteriophage. Okay, now they took this bacteriophage. This is your bacteriophage. It has a protein coat. Okay, it has a tail like structure. Okay, and this is the, so inside this what is going to be there? The genetic material, DNA is going to be there, and it was tested on an E. coli. Now, what it is going to do, the moment it finds a host, it is going to release the genetic material. Here what they took is, they took S35, radioactive substance. Sulfur is only present in protein. So, if protein is the transforming principle, if protein is the transforming principle, then we would have seen S35 inside the cell. Okay, because something is getting transformed means what? It is entering the cell, manipulating the cell. So, we took S35 as a radioactive substance to radio label the protein, which is not there in your DNA. Then we took P32, okay, P32, which is only present in DNA, not present in protein. So, this way we will be able to identify exactly who is the genetic material or who is doing what. What happened? No, Pradeep, just understand this way. When DNA was taken, okay, when DNA is degraded the DNA, the mice is going to live because DNA is the transforming principle. If DNA is not there, then it is not going to happen. Okay, so transformation where it is going to happen? Okay, no transformation is going to happen there. But see, what NCERT it is given, your transformation, it is just proven that DNA is the transforming principle. Okay, let me just open NCERT also. Let me see if any line is given there like that. One second. Just give me a minute. Yes, here. See, that DNA caused the transformation. So, here they are talking about, so I understood why you got confused. So, here DNA caused the transformation. Here and here transformation was in there. Pradeep, please read the statement here. Same thing what I have written here only is given in DNA, but I have taught you in a different way. That's all. Okay, because here, see in your NCERT what is written? They also discovered protein digesting and RNA digesting, okay, so that the transforming substance was not protein or RNA. Digestion with DNA did inhibit, okay, inhibit transformation, suggesting that DNA caused, okay, DNA caused the transformation. So, the statement is little, alag bulag here. So, that's why I give you this example because it's more easier to understand. Fine. Now go forward, S35 is only found in protein, P32 is only found in DNA. So when we are radioactively labeling it, we will understand that if it is S35, okay, labeled, and we are noticing inside the cell, then it is a protein, that is the transforming principle. But if it is P32, then it is DNA, okay. Now go forward and understand this, students. Bacteriophage is there, radioactively S35 labeled protein capsule is there. Now when we insert it in, obviously the first thing virus is going to do is what? Okay, first thing virus is going to do is what? It is going to put the genetic material inside. So the first ever transformation is going to happen is infection. First process is going to be infection. Okay, after infection, the protein coat is going to stay outside the E. coli. No, why do we want it, students? So we will do blending and we will remove, okay, blending or mixture, we will remove the protein capsules. Now, after that, we will do a process called as centrifugation. Centrifugation is a process where we take a centrifuge tube, okay, and we will rotate it at a different RPM, okay, RPM, and we will notice the heavier particle settling in the bottom and the lighter particle as the supernatant or the pellet. Okay, this, sorry, supernatant, the lighter particle, and pellet is the particle which got settled below, okay. Now, centrifugation happened. Look here, the result is very, very important. No radioactivity detected in the cell. That means what? Protein coat did not enter into the cell. That means protein is not the transforming principle. Where did we get all the protein? Where did we get all the radioactivity? Radioactivity detected in the supernatant. Clear to all of you? Okay. Here, radioactivity detected in the cell, that means DNA got inside, genetic material got inside the cell. So, DNA becomes the transforming principle. Here, no radioactivity detected in the supernatant. Is this clear? So, this is how Hershey and Chase proved that DNA is the genetic material. DNA is the genetic material. Any doubts so far on this? Okay, very simple, no, students. So, what did they do? They took a bacteriophage. Okay, they took a bacteriophage. Inside the bacteriophage, genetic material was there. So when it is going to infect a cell, okay, the next thing that it's going to do is it's going to release the genetic material inside the cell. After the genetic material is sent inside the cell, we will be able to notice if we have radioactively labeled this. So what we have done, let's say this is S35. S35 is only present in protein. It is going to radioactively label the protein. Okay, radioactively only going to label the protein. Correct? Protein coat is going to be labeled. This is going to be labeled. Correct? S35. Let's say we took P. Okay, we took P32. Okay, then the bacteriophage bacteriophage, genetic material is going to be radioactively labeled. Genetic material is going to be radioactively labeled. Now, I do not see any radioactivity inside the cell. That means protein is not the genetic material. Okay, and here I notice that this is the cell. Inside this cell, I notice radioactivity. That means genetic material is going to be DNA. DNA is the transforming principle. DNA is the transforming principle. Clear to all of you? So, 3 steps happen in Hershey and Chase. It is going to be first infection, where it is going to meet the host. Okay. Second is going to be blending. Using, we can use a mixer. Okay, here students, in this blending process you can use a mixer. Why? When you use a mixer, the protein coat will be removed. Okay, we use a mixer and with that help the protein coat will be removed, and the last process is centrifugation. Okay, so in the last process, remember that we can get a substance which is going to have a high, okay, which is going to be heavier. So this is the centrifugation tube. This is the centrifugation tube. We will notice that the pellet is going to settle at the bottom of the test tube because it is the heavier particle. Okay, it is going to be the heavier particle. This is the heavier particle. Then if we talk about the supernatant, then we are going to get the supernatant, that is this. Lighter ones is going to be on the top. Okay, lighter one is going to be on the top. Clear to all of you? This is the supernatant. This is the pillet. Clear? Pakka? Hershey and Chase experiment is clear? Avery, MacLeod and McCarty's experiment is clear? Any doubts so far? And please do the homework, okay? This is the homework question. Homework question has to come in the comments, okay? I want to see all of your comments about the homework. So today, that's it for today, students. Structure of DNA we have done, and all the experiments to prove that DNA is the genetic material has been done. And these notes are going to be available on the Telegram. So all these notes that I have written right now for you, which I have written before also, all of this is going to be available for you in the in the KCET channel. Okay, that's the Telegram channel, so definitely follow the Telegram channel. At the same time, if you want to join my Telegram channel, it is biology.
Okay, I found this on the web for Telegram channel at the same time if you want to join.
Did I say Siri by any chance? Okay, so students, biology by Gopika. Okay, this is the Telegram channel, and we Karnataka, KCET and boards. See, on this channel itself, if you go to this video, in the description itself, you will get the link. Okay, join the Telegram channel, click on that and join the Telegram channel. Okay, so all these notes will be available there, so do not fail to join the Telegram channel, and do let me know in the comments how the session was. And this is your homework question popping on the screen for all of you, and I want the answer in the chat box. Just put ABCD, I know the answer, so I am going to like the ones which are right. Okay, so heat kill S bacteria when inserted into the mice. What is going to happen in the mice? So see you all next time. Good night, take care, sweet dreams, and love you all. Take care very well of your health, and let's prepare really well for our competitive exams, and see you next time. Till that, tata, bye bye. Bega video like ಮಾಡಿ, share ಮಾಡಿ, friends ಜೊತೆ, ಆಮೇಲೆ comment ಮಾಡಿ. Okay.